Now is another very important code protein, which is one of the best understood, which is the clathrin code. So a lot of labs have really dissected this system very well. So it has the proteins that compose this clathrin code have been crystallized, and they know exactly the conformation of it. So it's really a very interesting system, which is what we call the triskelion, which is a combination of two proteins, a light chain and a heavy chain, that they arrange themselves in this kind of helix pattern, and they kind of fit around each other. As they assemble, it starts to give curvature to the matrix. So basically it forms this cage, which is always the same size, and you can see here the size bar. So this is larger than other vesicles, for example, the caviola that I showed you Tuesday. And the reason that the size is so constant is because of the angle of the assemble of these components of this cage. So this is how it looks on electron microscopy, right? So this is the type of electron microscopy in which we're just looking at the surface. The membrane of the cell was ripped open, and you're looking as if you were inside the cell. And you see these cages. So it kind of starts as a more flat assembly, and as the subunits start to join this patch, it starts rounding, and it forms this vesicle that then buds into the cell. So it has this very characteristic morphology that is very easy to identify on the electron microscopy, and it's very abundant because this is the type of code protein that really controls what we call constitutive endocytosis. So the endocytosis that the cell is doing all the time to bring important components.
Sorry, can you speak up a little bit?
Okay. So this is kind of a trick in this animation we're going to see, because this is transmission, right? Again, the sectioning type of electron microscopy, but they did something clever here, that they assembled a series of sections that is going to give the impression that the vesicle is budding. So this is how clathrin looks on transmission electron microscopy, right? You see this fuzzy coat, which corresponds to those proteins that we just saw the structure. And you can see how this progresses.
Eukaryotic cells take in extracellular molecules through a process called endocytosis, in which the plasma membrane invaginates and pinches off cargo-filled vesicles.
Right, so inside cells, sometimes you see structures like this.
This movie shows a series of
That really
electron
looks like it's one
that
that
have
just
artificially morphed
budded
to show the process of
the connection to the
endocytosis
membrane.
as it may. The process involves a variety of molecules, including the cargo molecules that the cell takes in, the receptors that capture the cargo molecules, and molecules called adaptins that mediate contact between the receptors and the clathrin molecules that act to shape the vesicle forming at the plasma membrane. In the electron microscope, individual clathrin molecules can be seen as three-legged structures called triskelions. Each triskelion contains 3 heavy chains and 3 light chains. When a clathrin coat forms on a membrane, the globular domains that make up the tips of the heavy chains bind to adaptins, which interact with cargo membrane proteins. The assembly of a clathrin coat can occur spontaneously as numerous individual triskelions come together, interact through their leg domains, and ultimately form a closed cage.
I swear I've seen this before.
Really for drama. I've seen this in scientific meetings that people put this to classical music all the time. So this has been very satisfying for a lot of scientists because it was a system that really was easy to study and to understand the structure. So there's many labs still involved in studying clathrin-mediated endocytosis. Other types of endocytosis are not as well understood. So this is the diagram of what happens, right? So this means you just saw the animation of the process. And again, the important concept in any form of budding of vesicles is that the cell has to have a mechanism to load proteins, right? So this means the cargo proteins, in this case here, would be the red dots, and receptors for these cargo proteins. So the key element that makes these proteins be directed to these buds is because the receptors for these proteins are recognized on the other side of the membrane, on the cytosolic side, right? So these cargo molecules would be outside in this case. The cell is just trying to take material in. This would be the plasma membrane. And you have on the cytosolic side proteins, again, the same concept. These proteins are floating around free in the cytoplasm. And when they meet a receptor, and we're going to see how this is regulated, they can bind to the tail of this receptor, and then they recruit the next components, right, which are these triskidial molecules that really shape the membrane. And this is how it progresses. So the process starts with the recruitment of the cargo molecule by the receptor. Yeah.
Wondering how do they have
They don't. So that's a very good question because in the beginning people were really rushing to crystallize it, and it was this big rush hoping that it was going to be something as satisfying as this, and it's not. And maybe the reason, that's why, remember that I mentioned that the COP2 can actually attach to larger buds to accommodate very large proteins. So in evolution, this seems to have been an advantage, not to be so well defined. But for this form of endocytosis, the main purpose of it is just to take smaller molecules out of the extracellular. So this is, again, the regulation, right? We're going through several examples here of how proteins that are in the cytosol get recruited, and then they start the process. So I'm going to introduce one more concept here, right? So we already went through the Rab proteins that are regulatory proteins that are Rab GTPases, and the proteins that form codes, and then the proteins that promote fusion. So another very important class of regulatory proteins are actually lipids. So among the phospholipids, right, that we know are very abundant on membranes, there is a class of phosphoinositol lipids in which they form these very important regulatory types of molecules. So you can see here, this is well understood in clathrin-mediated endocytosis, but this has been involved, this kind of phosphoinositide signaling mechanism, in many different membrane traffic events in cells. So in the case of clathrin-mediated endocytosis, the AP2 adapter it is an important protein, right? So this AP2 adapter is formed out of 4 subunits, and normally it is in the cytosol in this locked form. So this means that these subunits are not able to recognize the tail of the receptors. So it is the phosphoinositide in this form, PI45P2, that when it binds to one of the subunits of AP2, it then triggers the conformational change in which these proteins here suddenly can recognize the tail of these endocytic receptors. And that's when AP2 gets recruited to the membrane. So this phosphoinositide, this lipid on the membrane, is actually the initial trigger for this process. So this is a diagram, right, of how, what is really interesting about this is that it offers the cells many possibilities. So this is how it is the structure of this lipid, in which you have this sugar inositol group that has these hydroxyl groups that can be phosphorylated. So this here actually shows the whole collection of the different types of phosphoinositides that can be formed just based on how many phosphate moieties are added. So what these numbers mean is that between brackets is the position on the inositol ring in which the phosphate is added. And then this subscript here is how many phosphates were added into the original form, right? That it's only one phosphate. So this is really a very dynamic and versatile way in which the cells can very rapidly add or remove phosphate. And this is known to be an important signaling mechanism. So probably people here know, right? How would a cell rapidly remove or add phosphate? So let's talk first about adding phosphate. How can you add a phosphate group to a molecule?
Use ATP.
ATP is the source, right? But what do you need to do that in addition to ATP? How many people here have biochemistry? Kinase, right? Everybody heard of kinases? So kinases are critical, so there are many kinases that what people are discovering is that in each membrane that produces a specific type of phosphinosidide, there's specific kinase that can do exactly this type of phosphorylation. Right? So what is the enzyme that does the opposite reaction, that removes phosphatase, right? So there's a big hunt right now. It's still very much in the beginning, so this means the whole discovery of these single lipids is, I think, less than 10 years old. So people are still looking for the kinases and phosphatases. But what is emerging is that they are kinases and phosphatases strategically located on the membranes in which these different forms have to be produced. So this is, again, right, so this means this is a larger representation of how these soluble molecules, right, that can be 82, for example, in the case of collagen-mediated endocytosis, but there are many other types of proteins that can be recruited from the cytosol, and they can recognize specifically the number of phosphates on this inositol group. So this is why it's possible to distinguish, because different proteins get recruited depending on the degree of phosphorylation of these lipids. And as what we looked before, right, the big advantage is that these lipids are actually, they can move on the membrane. So they can really be easily recruited to the sites in which this is needed. So this is another example, right? So we already saw that there are specific snare molecules, the proteins that promote fusion, that serve as address tags of different stations inside the cell. Then we saw that RAB, the RAB GTPases, which helped with the tethering of the vesicles, they also are localized, and you actually can guess what is the organelle you're dealing with just by looking which RAB is there. So this is one more example that very interestingly people are discovering more and more, that different compartments and importantly even different domains. So you can see here, for example, that this budding out of the trans-Golgi, you have some forms of phosphinosides that are only at the buds. While if you look at endocytosis, you have another form, and if you look at large objects that are taken in by phagocytosis, you mostly see PI3P. And a big advance was to develop reporters. So basically, cells can express proteins like these proteins that we saw before that recognize specific forms of these phosphinosides. And they tag them with fluorescent markers, so you can actually see the recruitment of these lipids and then going away. So I think I have a movie here. This is actually a movie from my own lab, in which we were looking at a cell taking up a parasite. So this is a trypanosome, an intracellular stage of the parasite that can also be phagocytosed. So this is a macrophage that is going to take up this parasite, and you're going to see what we are using as a reporter here is this 2,5-GFP. So 5 is a protein that recognizes specifically PI3P. So you're going to see that as the parasite starts getting engulfed, that it gets really intensely recruited and then stays there for a while and it falls off. Oops, so what happened here? I didn't want to do that. I'm running projection mode, sorry. I play is falling into the, seems to be an iPad glitch. Oh well. You're gonna see the phagocytosis only in phase contrast, which is a little problem. The important thing is that, why don't you look at it later online? So at this stage here, in which the parasite was completely engulfed, you see a very intense green fluorescence that stays there for a while and then falls out. There's some problem here that when I play, it's only playing one of the movies, not the second one. So another, we touched on this briefly on Tuesday, but I didn't show you how this works, is, right, we talked a lot about how these coat proteins can deform the membrane and make it pinch off. But it is really strongly believed that this is not enough, just this conformational change of the membrane. And you need a specific protein that pinches off this neck of the vesicle. So again, in the case of clathrin-dependent endocytosis, because it's such an easy system to study, people found this GTPase, again, it's a GTPase dynamine that formed this coil. It can even be seen on electron microscopy. This was first discovered in a screen in the fruit fly, actually. And this is a picture of clathrin-coated vesicles on a fruit fly cell showing these rings on the neck, which correspond to this protein. So this regulation by phosphonosidides, in this case, is PI45P2, that it has a binding domain that recruits the GTPase, activates basically the GTPase domain that then promotes the pinching. So this is a representation of how these chains of dynamine would wrap themselves around the neck of the pinching vesicle. So it's not clear if there are other types, so this means mitochondria have kind of a similar protein that is involved in dividing the mitochondria, but there's many, for example, the COP2 vesicles, we don't know yet exactly what is the pinchase, if there is one. So there's still a lot to be discovered in this field. So I told you that this clathrin-mediated endocytosis is one of the most important forms of endocytosis, right? And there's one example here of one form of endocytosis that is extremely important, which is low-density lipoprotein that carries cholesterol. So basically, the receptors for LDL that are on the plasma membrane, they are recognized by clathrin coats. And then these particles of cholesterol with the low-density lipoprotein are brought into the cell. And this has been extensively studied and was one of the ways in which people really worked out what we call the maturation of the endocytic pathway. So what happens is that you have these coated vesicles, then the coats fall out. You have then a naked vesicle that keeps moving in, and then it joins these more irregular structures, the early endosomes. And this is very dynamic because there's vesicles constantly budding out of this and returning to the plasma membrane, right? Again, that concept that you have to equilibrate the amount of membrane in each station. And then there is what we call maturation, that these compartments, they get gradually acidified. We're going to look into this. And they receive enzymes, hydrolases from lysosomes. So you have these intermediate stages that we call late endosomes, and then the digestive vacuole that contains the lysosomal enzyme. And so the LDL particle that is up to here, complexed with cholesterol, it's degraded at this point. The cholesterol is released, while the receptor can be recycled back to the membrane. So the cell really uses this a lot, in which to reuse the receptors. So the receptors don't go so far, so they're not degraded. Right? and the low pH of these compartments, when it starts to acidify, it releases the cargo from the receptor. Okay, questions? So let's look at one more animation, which this shows very nicely what has been learned about the endocytosis of cholesterol through LDL.
Cholesterol circulates in the bloodstream and then enters cells by receptor-mediated endocytosis. Instead of circulating freely, cholesterol molecules are derivatized and packed inside low-density lipoprotein particles, or LDLs. A protein and phospholipid layer surrounds the cholesterol molecules. The protein portion is recognized by LDL receptors on the surface of cells. An adaptor molecule, called adaptin, binds to the tail of the LDL receptor that protrudes into the cytosol. Adaptin recruits clathrin molecules, which start coating the membrane. Assembly of the clathrin coat causes the membrane to bend and invaginate, forming a vesicle that buds off inside the cell, taking with it LDL receptors and the LDL particles bound to them. Once inside the cell, the vesicle uncoats and fuses with the endosome, the intracellular compartment that first receives all endocytosed material. The endosome has low internal pH, which causes the LDL receptors to release their cargo. Empty LDL receptors are recycled to the plasma membrane in vesicles that bud off from the endosome. Each LDL receptor makes a round trip from the plasma membrane to the endosome and back every 10 minutes. Meanwhile, the LDL particles need to be disassembled. The endosomal content is delivered to a lysosome, which contains hydrolytic enzymes that can digest the particles. Free cholesterol is liberated, together with amino acids and small peptides generated by digestion of LDL proteins. The cholesterol is then released into the cytosol to be used in the synthesis of new membranes.
So of course, right, you know how important this pathway is. So I wanted to give you some details of this management of cholesterol. So you hear a lot about this, right? It's in the news all the time. There's the bad cholesterol and the good cholesterol. So low density lipoprotein is called the bad cholesterol. And the reason that it's called bad cholesterol is because when the receptors get saturated, it can remain in circulation. These receptors are really very important to really keep pulling LDL out. So if the receptors get saturated, if there's too much LDL in circulation, that's when this deposition in arteries can start to happen. Right? So this is the whole origin of the whole discussion if the diet really rich in cholesterol is dangerous or it's not dangerous. So, and the balance between the LDL and HDL is pretty important because the high density lipoproteins, which are called the good cholesterol, because they have a different mechanism in which they can really basically steal the cholesterol out of the LDL. So you have a lot of LDL complex cholesterol in circulation. HDL, if it's in higher amounts, can take this cholesterol and then take it to the liver. So removing it also from circulation. So this is a very useful diagnostic tool, right, for risk of heart disease, the balance between LDL and HDL. And then there are these drugs, right, the statins, that millions of people are taking this. So how does this drug work? It's an inhibitor of this reductase, HMG reductase, which is a critical enzyme in the pathway for making cholesterol in the liver, right? So cholesterol doesn't only come from the diet. Actually, the most important source of cholesterol is the synthesized food. So this is what gives rise to all this discussion. So is it really important to stop eating cholesterol-rich foods? Does this make a difference? Because if you are synthesizing too much cholesterol or not clearing it properly, it's not going to make much of a difference. So the mechanism by which they discover that this drug works is that they inhibit the synthesis. So then, of course, the levels of cholesterol are going to fall. But the interesting side effect is that when the cell sense that there's low levels of endogenous synthesized cholesterol, they start making more LDL receptors. So basically, the two things happen. You shut down the production of cholesterol, and you start putting more receptors on the membrane that can pull in the circulating cholesterol. Right? But it's important to understand this because this is a major alteration of cellular function that these drugs are doing. And people take it for life, right? So it looks like it's true, it's pretty safe, but it's important to be aware of what is happening exactly, right? So this means you're basically interfering with two major mechanisms.
Is there any major side effect of going out for LDL receptors?
That's a good question. I'm not aware if it's understood completely how the receptors for HDL is a much more complicated process. I just don't know. We could look it up. It's something that LDL has kind of, uptake has kind of been the workhorse in a lot of serology labs, so they know the details of it. And I think HDL still has to be worked out. Okay, so let's come back to this concept, which is another important cell biological concept, right? We have been talking about the topology inside compartments. This corresponds to the outside medium. But one important exception is that cells place on these membranes. So in this case, let's say that this is an endosome, or actually already a lysosome because the enzymes are here. So this VAPPase, which is this very important proton pump, which we saw as the energy-dependent transporters that really can, using the energy of ATP, translocate ions against their gradient. So actually there's much more of these protons kept inside the vesicle than outside. So this is an active pump. And this is different from the outside medium, right? So the outside medium is neutral pH, and inside these endosomes, starting on the early endosomes and then getting more and more acidified. And it's a very interesting story that is well understood that reflects how many of these pumps are added to the membranes. So as these endosomes mature, they get more of these pumps and they acidify more, to the point that when it gets to the end, the lysosome, the pH inside is something like 4 or lower. And this is a pH that really, most of these enzymes that reside in the lysosomes, they work best. So this was a mechanism to, if these lysosomes break and these enzymes spill out, the cytosol is neutral pH, outside the cell is neutral pH, so these enzymes can't do too much damage. We know that they keep working for a while, but this is one mechanism. So this is one thing to keep in mind, that yes, the lumen of organelles is topologically equivalent to the outside, but in the endocytic pathway, the cells change the internal pH and make it acidic. So another clever mechanism that is used to take enzymes to lysosomes, right? So what we're looking at here is again the Golgi, and we already saw this, that this processing of the glycosylation happens in different stations of the Golgi. So what was discovered is that this mannose-6-phosphate, so it's a terminal phosphate group that is added while the protein is traveling through the Golgi. So when this group is uncovered, it finds a receptor. There is a receptor located in this trans region of the Golgi that can bind to mannose-6-phosphate receptor. And this turned out to be a really exciting discovery because people did not understand properly how do you get the proteins from the lysosome to go only to the lysosome. And it turns out that this mannose-6-phosphate receptor functions as a shuttle. It kind of takes it only to these endosomes and then it recycles back. And it keeps transferring these enzymes. And some of this receptor actually can be on the plasma membrane, so it can recover enzymes that by mistake are sent outside the cell. And then this works in the same way. When these vesicles containing the receptor coupled, say that this is a protease, a lysosomal protease that is glycosylated, it has mannose-6-phosphate receptor, it arrives in the lysosome, and the low pH separates it from the receptor. So the receptor is free to go back and be reused. And then this enzyme finds itself in its environment that it can work. Right? And there are many fascinating mechanisms that we don't understand completely yet. These enzymes are basically not degraded by other enzymes. So they actually are protected from degradation inside the lysosome, and they're basically waiting for material to be delivered. There's more than 50 different enzymes that are specific for lysosomes. So this is a closer view, right? What is the mannose-6-phosphate modification? So you again have a mannose with a phosphate in the position 6. And this is a terminal group that is added to this N-linked oligosaccharide that we looked at before. This is the basic glycosylation unit that is added on the cytosolic side and then is flipped in. So when it's flipped into the YAR, it can move to the Golgi, and in the Golgi, this mannose-6-phosphate is added. Only if this is a protein that is destined to lysosomes. So on a electron microscopy, again, the story of lysosomes really has a long history going back to the same time in which people, Claude and Paladi and Ledoux were the three that got the Nobel Prize for membrane traffic. And they already knew that these organelles that they called lysosomes had acidic hydrolysis because they developed very clever mechanisms that you have these slices of cells and you can do a reaction. You put a substrate that gets cleaved by these enzymes. And even in fixed preparation, some of these enzymes are active enough to basically create this reaction here. So basically precipitates something that is electron dense and then they can be seen. So this acid phosphatase was one of the enzymes that they managed to develop this. So this became a very widely used reporter to people to say what is a lysosome itself. Organelles that contain acid phosphatase. And then what emerged is that there is a related organelle, right, which has a lot of these small vesicles inside, which we now call this multivesicular body. And we know that this is in the pathway of becoming a lysosome, because the way this works is that membrane from the outside membrane of these compartments starts budding into the lumen. And this is a mechanism in which the cells use. For example, if the cell decides to reduce the level of receptors, right? Instead of the receptor recycling back to the plasma membrane, it can bud into the lumen, and then the enzymes are there aiding, waiting to chop it up and degrade it. So these vesicles that we see on electron microscopy is actually membrane from the outside membrane of the lysosome that is budding in. So until very recently, nobody knew how this worked. And looking at this system, now it's known that there is another cytosolic complex of several proteins in the cytosol. They get recruited, and it's called the escort complex. And interestingly, the way people first discovered this was looking at viruses. So it was groups working with HIV that they saw that when these viruses, right, because viruses, they take over the machinery of the cell, and they basically use the membrane of the whole cell to form its capsule when it buds out. So they discovered that this group of proteins here, especially escort 3, they can form these spirals that constrict the membrane, and this has an opposite topology. So the important concept here is that this is exactly the opposite of a Clathrin coat, for example. Clathrin coat is on the cytosol pulling the membrane in. And this escort complex, nobody completely understands. There's very interesting discussions going on. But somehow this protein, when it assembles, it pushes the membrane out, and it makes these vesicles. So viruses use this, and the second process that people discovered that uses this is the budding of membrane into the lumen of lysosomes. So this is mediated by this sequential deposition of escort 0 is the one that recognizes, again, the signal here is an inositol phosphate, which then recruits receptors that have ubiquitin modifications, and this triggers a cascade. It keeps recruiting other components until getting to the last one that is able to deform the membrane. So the only thing to remember is that there is a kind of an anti-coat in which there's a complex of proteins that pushes membranes in the opposite topology, and this is used in the maturation of the lysosomes. So this is the classical diagram, right, of the maturation of endosomes. This is another area in which there was a lot of fights and discussions between some biologists, because one of the discussions was kind of similar to that discussion between the Golgi, cisternal maturation, or vesicle shuttling. So the discussion was, are these stations, the early endosomes and the late endosomes and the multivesicular bodies, kind of fixed in cells, perhaps anchored in the microtubules, and then vesicles just shuttle stuff between them? So this kind of has fallen out of favor, this idea. It's pretty clear now that these irregular endosomes, they form, they break apart, and they form again. This is a constant flow. But it is true that they are associated with microtubules, and this is a system that keeps pushing all these membranes deeper into the cell until they meet the lysosomes. So it's really more or less a consensus now that this is a maturation process. This is not a transfer between pre-existing stations in the cell. Although at some point people believe that. So just to mention, focus a little more on lysosomes, right? So what is really very interesting, I'm kind of biased, in my lab we work with lysosomes. We think it's really the most interesting organelle because it is at the center of many, and more, it used to be that people thought, well, it's just a boring garbage can. That it basically receives the material that the cell is taking up from the outside and then degrades. And then it just transfers to the cytosol, the units, the amino acids, and nutrients. What is emerging is that it's much more than this. That lysosomes, for example, they are very important in defense against pathogens, for example, right? So phagocytosis that takes up larger particles, including many pathogens, also meet lysosomes. Some pathogens block this fusion with lysosomes. Other pathogens inhibit the action of the lysosomes not to be killed, and we're going to look at this in a later lecture. And the other very fascinating process that emerged that was known for a long time based on electron microscopy, but now finally, again, using yeast genetics, that we are beginning to understand how autophagy works. So there is this mysterious mechanism in which these double membranes, right? So they kind of look like an ER membrane that they can be seen engulfing certain organelles in the cytoplasm. And then at one point they form this complete enclosure, and somehow these two membranes become only one membrane, and then it fuses with the lysosome. So this is a very important process that cells use not only to turn over kind of defective organelles. For example, when mitochondria are getting old, they have to be recycled. This is one mechanism. But the cells tightly regulate this. And when they are low in amino acids, right, so we give a starvation response to cells, we take amino acids away from their culture medium, you can get the cells to start digesting itself, start eating its own organelles. It's something that is very easy to regulate. And it turns out, just in recent years, that this is controlled by this this complex, mTORC1, which is, again, a cytosolic complex that is recruited to the surface of lysosomes, and it functions in a cascade of phosphorylation. And it is a sensor, and this is still being worked out. Folks still don't have exactly how this works. The only thing we know is that there's a sensor for the concentration of amino acids. When it's low, the cell then inhibits this complex, and then autophagy starts to work. So it's basically a regulator of autophagy. And people think that it's actually much more than this, that it's a major hub of controlling the cell metabolism. So you should keep an eye that this means there are papers coming out every week on something else they discovered about mTOR. So this is an image of an autophagosome, a classical one that contains a mitochondria, right? Very easy to recognize here. And this is a peroxisome, another organelle that is easy to recognize because of these crystals that it has inside. And you can see that the membrane around it, it's two bilayers. And this is the classical image of an early autophagosome. So there's still a big debate. Some people think that this membrane comes from the ER. It has been suggested that maybe it comes from mitochondria. So membranes are recruited and are folded around organelles. And this is the beginning of the process that is going to ultimately deliver this to lysosomes for degradation. So this is the diagram here. You have this membrane that folds is basically a tubular membrane, a compartment of two membranes that engulfs the organelles, and then it is believed that very soon one of the membranes disappears, and this is believed to happen because the lysosomes bring the hydrolases in and start eating up this internal membrane, fragmenting it, and also then degrading the organ. So that's autophagy. And this is another process in which, just to mention briefly, that I told you that clathrin-coated vesicles are very uniform in size, but cells have another way of making larger vacuoles. And this is called macropinocytosis. So this is a mechanism that is controlled by polymerizing actin. So it's similar to phagocytosis in the sense that bundles of actin, you're going to see in the next class, this drives the membrane protrusion, and then this folds on itself and then emerges, forming a large vacuole. So this is very common in many cell types, and coupled to big roughings of the cytoskel. So I just wanted to mention again, right, to make links with diseases, Parkinson's disease, right, a very serious neurodegeneration disease. So it has been worked out not long ago that there are mutations in these two proteins, PINK1 and PARKIN, and they are enzymes that really recognize damaged mitochondria. So PINK1 basically brings the enzyme that can add ubiquitin to damaged mitochondria, and then this ubiquitin addition is one of the signals to engulf and degrade these organelles. So people now understand that Parkinson's disease is associated with a defect in autophagy, at least one of the genes that can lead to Parkinson's disease. So another concept is right similar to what we already discussed. When you get at the trans-Golgi, there's all this traffic going on, but the cell has to distinguish what's going to go where. I already went through the mannose 6-phosphate receptor, this very clever mechanism that uses a sugar tag to identify lysosomal enzymes. So these bud in a specific region of the trans-Golgi, and it goes to lysosomes. Then you have what Jim Rothman actually, I call this bulk flow. So the idea is that whatever is not selected specifically to be packaged into specific secretory vesicles would keep moving out. So this is the idea here, that you have constitutive secretory pathway because you have to deliver membrane proteins, at least the plasma membrane all the time. Right? And the last important concept that we didn't talk about yet is regulated exocytosis. So there's a lot of cells that basically form these secretory vesicles, that they store a lot of material, but it's not good for the cell to be constantly putting this out. So this is held back, and a signal is needed to really make these vesicles fuse. So this was another very active area of research. People knew that calcium is a very common signal, right? Calcium very low in the cytosol, when the calcium goes up, many secretory granules can start to fuse. But how is this controlled? Because we know that all these membranes have snares. If they meet the membrane, they should fuse. So what emerged, right? This is just showing a closer look of how these secretory vesicles are formed. And there's a concentration of these secretory proteins in these vesicles that many times they just get stored and they sit there in the cell until the signal comes. So there's a cargo concentration process going on. And this, I just want to mention briefly that this type of regulated exocytosis has emerged recently to be very important for fundamental mechanisms in cells. When cells divide, for example, now that we have such good imaging methods and we can basically peg membranes and watch what they're doing, when the cell is ready to divide, there's actually addition of membrane that actually increases the cell area immediately before these two daughter cells separate. Phagocytosis is another process in which we know now that regulated exocytosis of small vesicles kind of gives more membranes to form these large vesicles. once the embryos are forming cells, they need a lot of membrane, right? The process that we call cellularization. There is also quite a bit of evidence that vesicles are recruited from the inside, and they fuse in a regulated way only when it's needed. And then this is dear to my heart. This is something we work in my lab, and this was something very gratifying because Alberts mentions very briefly that these vesicles that fuse in wounded cells have been identified as lysosomes, and this happened in my lab. So, to our surprise, the lysosomes get the signal from a rupture on the membrane, and they fuse to help with the receiving process. So, at least one little part of one little figure, we made it into the textbook. But what I wanted to talk to you mostly is, of course, what is more understood in detail, and we come back to the nerve terminal. Right? So this means you have this very vigorous cycle of synaptic vesicles full of neurotransmitter, and neurons are unique in which they can have these very long axons. So one of the problems that people were interested in is how you refill these or make more synaptic vesicles if the cargo has to come all the way from the Golgi, right, in the classical pathway of traffic. So it turns out that in synaptic vesicles, there is a way to really make more of these vesicles in a faster way. So you have here these early endosomes that are usually closer to the plasma membrane, and these vesicles that fuse to the membrane and release the neurotransmitter can be refilled and not have to go through the whole process of having to go all the way from the Golgi. Right? So what would you think that is the mechanism? Why is it possible to do this in the case of neurotransmitters? To do this easy, fast refilling of vesicles? Which mechanism cells are using? There's people that I never look at because I'm always on them. Any suggestions? Natalia? How do you put something inside across a membrane if you're not using the mechanism that we saw that you translocate across the ER using that complicated translocome mechanism? What is the other way that cells can transfer molecules across membranes? Very? Channel proteins. Yeah. So what Natalia was going to say?
A transporter maybe?
Exactly. So same thing, right? The transporters or channels. So the key thing here is that neurotransmitters are very small molecules, right? So this means transporters, subtransporters, the ABC transporters, for example, can deal with small proteins, but it has to be small. In this case here, there are channels and transporters that can really put these neurotransmitters back into the vesicles. So basically, after they delivered into the synaptic space their content, they merge back with these stations, and then they are recruit the transporters to that bud, and then the neurotransmitter that is in the cytoplasm can go back in. So it's more complicated than this. Of course, everybody knows that the two mechanisms, there's also traffic coming from deeper in the cell, but there is a fast recycling of synaptic vesicles that is very important for the efficiency of the process.
So we know that there's also an uptake of the neurotransmitters called signal stones. Is that through channels? Or is that through axis? They go through axons?
Right. So when outside, right on the next cell event terminal.
Well, no, once they release their neurotransmitter signals, that they re-uptake the neurotransmitter.
This can happen, but since this is with the synaptic cleft has the receptors for the neurotransmitter, a lot of them get captured on the next cell, so it's mostly what the next cell does with it.
Okay.
But I believe that in every mechanism in membrane traffic, there's that too. There may be some reuptake, but it's not thought to be the major mechanism by which they refill, because there's going to be competition with the receptors for them on the next cell, which are high. This is really what is the very interesting concept that in the cytoplasm, it's kind of a luxury environment that these molecules can just stay floating over there and kind of not be removed until a signal makes them be recruited to a certain membrane. So the cytosol acts as a more efficient storage of a lot of molecules because they are basically idle there until they are called into action. So I just want to mention again, right, so this means introducing one more element on this snare-mediated fusion. So we talked a lot about this, that this was the real major breakthrough that gave Jim Botman the Nobel Prize just 3 years ago, in which these membrane-anchored snares, one on the acceptor membrane and another one on the donor membrane, combines with a third one that is only loosely attached. This forms this tight bundle that is believed to provide the force to really push the membranes together and get the fusion. So the third winner of the Nobel Prize in 2013 actually was Thomas Sudoff, which had for many years worked on this protein synaptotagmin. So synaptotagmin is a calcium sensor. So it has these two domains that really bind calcium. So what he showed in many different types of experiments is that it is this molecule, the synaptotagmin, that is also residing, so it's on the membrane of synaptic vesicles. He comes from a group of biochemists that what they did for a long time was to purify synaptic vesicles, that you can get that in large amounts from the brain. And he found that there was on the membrane of these synaptic vesicles, these proteins that had two domains that can bind calcium. So they showed with mutations and many different types of experiments that it's not part of the snare complex, but if you have synaptotagmin on the vesicle, the synaptotagmin, after binding calcium, facilitates formation of the snare complex. So this is the concept of how do you do regulated secretion, that you have molecules that are very close to the fusion machinery that can tell, well, the calcium is up, so I should stimulate this process, right? So it's what we call calcium-regulated exocytosis. So just to show you here these synaptic terminals, how they are absolutely crowded with proteins. So this is an attempt, they did a very nice one in the textbook of really giving you an idea how crowded these cellular environments are. They try to draw these things to the real known number of proteins, and especially the synaptic vesicles, because they have purified them. They know how many of each molecule is present. So I have a little animation here, which is going to discuss the proteins in synaptic vesicles. So this is drawn to scale as close as possible.
This model represents a cutaway view of a synaptic vesicle, with the membrane lipids and proteins drawn to scale. Each synaptic vesicle membrane contains approximately 7,000 phospholipid molecules and 5,700 cholesterol molecules. Each vesicle also contains close to 50 different integral membrane protein molecules, which vary widely in their relative abundance. The most abundant protein is a snare protein, called V-snare. This molecule participates in membrane fusion at the synaptic terminal, and there are about 70 copies of this protein per vesicle. By contrast, the vesicle contains only 1 to 2 copies of V-ATPase. The V-ATPase uses energy from ATP hydrolysis to pump protons into the vesicle lumen. The resulting electrochemical gradient provides the energy to import neurotransmitter molecules, such as glutamate, into the vesicle. Through a transporter in the membrane, protons flow down their concentration gradient to the outside of the vesicle, as glutamate enters by an antiport mechanism.
Okay,
In this way, a vesicle is loaded with thousands of glutamate molecules. This model was created in part from an electron tomogram of a real synaptic vesicle. The tomogram combines visual slices of the vesicle at various angles to create a unified image. Other data, including structural data of the synaptic vesicle's proteins, have been combined with the data from the tomogram to create the three-dimensional model on the left. Note, only 70% of the membrane proteins estimated to be present in the membrane are depicted in the model. A real vesicle would be covered by an even more dense forest of proteins.
they tried to draw everything and they couldn't fit it, so this is 70%. Okay, so what I would like to do with the last 15 minutes, so this means in this case, I want everybody to do it on their own, but you're free to consult your notes. So just 4 questions, doesn't have to be a long answer, but conceptual things. So just write your name on it when you're done.